Hello, everyone. Welcome to our 5th episode in webinar series from Biochemistry and Biotechnology series in the UNIgreen Alliance. Today, our topic is about advances in plant molecular physiology. Before I give the stage to our speaker today, I would like to give you a few information about UNIgreen, if you are not familiar with our alliance. I'm Patrick Gonzalez. I will be the moderator today, and I'm co-coordinator of research and innovation for the UNIgreen University. As a disclaimer, this event is recorded and it will be uploaded in our UNIgreen YouTube channel, where you will find also our previous webinar. If you are a student, you need a certificate, don't hesitate to write us at this address here, and we will make a certificate of attendance for you. And of course, please feel free to use our chat to ask the question, and we will come to your question at the end of the webinar. So about UNIgreen Alliance, so we are 8 universities across Europe. This is an alliance that is supported by Erasmus Plus program, and we dedicate UNIgreen to research and education in the field of sustainable agriculture, green technologies, and environmental science. So we are taking both aspects, education and research, and we have a lot of opportunities that are being created around UNIgreen. So stay in touch, come to our website, and you will see all this opportunity, and don't hesitate to candidate for the different workshop and courses we are offering. So within UNIgreen, we have 8 axes of research that are listed here. And today we are in the first axe of research, which is biochemistry and biotechnology, but you can see that we are covering much more aspect from within this field. So as I mentioned, this is our 5th webinar. You will find all the other webinar on our YouTube channel, but also on the website. So I give you the links here. So today we are within the topic advances in molecular plant physiology, and in March we will have a subject about food-derived bioactive substances and their interaction with health. So stay in touch for the next webinar. As for today, we are really happy to welcome Professor Gawrownski, who is Associate Professor at Warsaw University of Life Science, and he is working in the Department of Plant Genetics, Breeding and Biotechnology. So today we will have really the chance to know more about one fascinating organelle, which is chloroplast, and more particularly we will be diving into the translation within chloroplast under abiotic stress condition. So Piotr, we are eager to hear from you. So I will give you the stage now.
One more time, thank you, Patrick, for a nice introduction, and good morning, everyone. My name is Piotr Gawrownski, and today I will talk about how chloroplast translation is regulated during acclimation to high light conditions. I will focus on translation because this is where chloroplast can respond very quickly to diverse stress conditions. And I will start the presentation with the biological context, then show the PSBA gene as a model of the translational regulation, and finally extend this to elongation regulation, transfer RNA biology, and new RNA sequencing approaches. But before we dive into the main topic of the presentation, let me briefly show you where the Warsaw University of Life Sciences is located. So our university is situated in the southern part of Warsaw, and the entire campus is concentrated in one well-equipped area. And our department, the Department of Plant Genetics, Breeding and Biotechnology, which is the part of the Institute of Biology, is shown in the top right corner of this photograph. Now returning to the main topic of this presentation, as you probably know, plants are constantly exposed to changes in light intensity. And when light becomes too strong, it is no longer beneficial and instead becomes a stress factor. As a result, plants respond to excess light at many levels, including leaf movements and chloroplast movements, and, for example, non-photochemical quenching, which dissipates excess energy as non-destructive heat. Moreover, highlight response is often linked to the redox and ROS signaling, which in turn leads to metabolic and enzymatic changes. One of the visible examples of this response is the accumulation of the anthocyanins, as you can see in this photograph, in Arabidopsis plants exposed to highlight stress. Of course, highlight also induces extensive changes in gene expression. However, even with this, all these protective mechanisms in place, highlight can still damage the photosynthetic machinery. And photosystem 2 is the most light-sensitive component of the photosynthesis. And under highlight, excess excitation energy leads to the formation of the reactive oxygen species, ROS in short, especially the singlet oxygen that is being generated in the proximity of the photosystem 2, the PS2 in short. In turn, singlet oxygen causes damage to the PS2, which becomes inactivated. This damage is, for example, reflected by the decrease of the chlorophyll fluorescence parameter, FV over FM, indicating the reduced PS2 efficiency, as you can see here. However, as we can see in this plot, after some time, the FV over FM returns to its normal level, reflecting the repair of the PS2, which is an integral part of the acclimatory response to the highlight stress. The primary target of the PS2 photodamage is the D1 protein. D1 is a core reaction center protein located at the heart of the PS2, and notably it's encoded by the PSBA gene, which is the plastid gene located in the chloroplast genome. Under highlight, D1 is damaged and degraded within minutes. So this is a very fast reaction. And notably, the D1 has one of the highest turnover rates among all proteins in Arabidopsis. Crucially, to maintain the photosynthesis, damaged D1 must be continuously replaced by the newly synthesized protein, and this makes this replacement directly dependent on the chloroplast translation. Here you can see highlight-induced PSBA translation activation in tobacco plants over time, in plants exposed to the highlight stress after some time. This analysis was performed using the ribosome profiling that allows us to analyze the translation efficiency. So we can see the induction of the translation efficiency of the PSBA gene, leading to the generation of the D1 protein in tobacco plants. And we also observed similar responses in Arabidopsis. And other labs, including the Professor Alice Barkan lab, reported comparable results in maize. So based on these observations, we can conclude that highlight-induced activation of the PSBA translation is a conserved response to highlight stress across plant species. In chloroplast, gene expression is regulated differently than in nucleus. The plastid genome is small, polycistronic, and transcriptional regulation is limited. And chloroplasts do not import transfer RNAs and ribosomal RNAs, so gene expression must be regulated locally. And as a result, the translation is the main regulatory step of the chloroplast gene expression. Chloroplast translation is catalyzed by the bacterial type 70S ribosomes. These ribosomes synthesize about 80 plastid-encoded proteins, including one of the most abundant protein on Earth, RBCL, large subunit of Rubisco. And translation in plastids relies on about 30 transfer RNAs, tRNAs, that together can decode all 61 codons in the mRNA. And chloroplast translation can be regulated at two main stages, initiation and elongation. And first, I will focus on translation initiation. Translation initiation in chloroplast resembles bacterial initiation. It depends on cis elements in the mRNA, such as Shine-Dalgarno sequence or the local mRNA secondary structure. These cis elements are located upstream of the start codon and allow proper positioning of the ribosome during translation initiation. The Shine-Dalgarno sequence, shown here in blue, base pairs with the 3' end of the 16S ribosomal RNA, shown in green, known as the anti-Shine-Dalgarno sequence. The 16S, of course, is located in the small subunit of the ribosome. This interaction secures correct positioning of the ribosome during the translation initiation. On the other hand, local mRNA structure works slightly differently, and it ensures that both start codon and the upstream region are in the open single-stranded conformation, allowing the ribosome to bind effectively to the mRNA. And some plastid genes rely strongly on Shine-Dalgarno, while the others depend more on the RNA structure, which is also similar to, for example, regulation of the translation initiation in E. coli. However, this makes translation initiation an effective control point for rapid responses to environmental changes. The PSBA mRNA is a classic example of the translational regulation in chloroplast. And on previous slides, I showed already that the highlight conditions induce the translation of the PSBA mRNA. And this response supports the rapid E1 replacement in these conditions. To study this process at the molecular level, we combined RNA sequencing with the ribosome profiling called RiboSeq to measure the translation efficiency. And we also used two techniques, DMS MapSeq and ShapeSeq, that probe mRNA structure in vivo. And our data show that highlight leads to the remodeling of the PSBA mRNA near the translation initiation region. Specifically, a stem loop structure observed under low light conditions is melted under the highlight conditions. And this structural change increases ribosome binding to mRNA and activates translation initiation. Thus, highlight activates PSBA translation via mRNA structure-dependent initiation. We further hypothesized that this light-dependent change of the mRNA structure is caused by the binding of the regulatory protein to the 5' UTR of the PSBA mRNA. But right now, the identity of this protein is unknown. Another layer of the translational regulation operates at the level of elongation. And translation elongation does not proceed at a uniform rate in many organisms, To study this process in chloroplast, we also implemented the ribosome profiling using the ribosec, called ribosec. Our data show that translation rates in chloroplasts are likewise non-uniform along plastid mRNA. And here, for example, you can see the example of the PSBC, another plastid gene with the ribosomes positioned on this mRNA. As you can see in this plot, ribosomes pose at defined positions, which appear as peaks during the elongation. And we found that these poses are linked to the mRNA structure, to the integral Shine-Dargano-like sequences and features of the nascent peptide chain that is synthesized by the ribosome. Importantly, these pose sites are conserved and functionally relevant. For example, we showed that ribosome posing facilitates efficient insertion of the transmembrane proteins into photosynthetic membranes. Okay, and so far I have focused on ribosome and the mRNA, and the next question is how the transfer RNA, the tRNAs, can contribute to the regulation of the translation. And chloroplasts contain a reduced set of about 30 plastid-encoded transfer RNAs. These tRNAs decode all 61 sense codons using mechanisms such as wobble and super wobble base pairing, which allow relaxed pairing between the tRNA and mRNA. And because of this reduced tRNA set, chloroplast translation is highly dependent on tRNA properties and availability. The tRNA molecule has a conserved secondary and tertiary structure. In the secondary structure, we can distinguish key functional elements, which include anticodon stem loop, D and T arms, and acceptor stem, which binds the amino acid. The tertiary structure of the tRNA resembles L letter, and the structure fits well to the ribosome. tRNAs carry multiple post-transcriptional chemical modifications at the nucleotides, including methylations, thiolations, isomerizations, and other complex modifications. And these modifications cluster at the key functional regions of the tRNA, including the anticodon loop and D and T arms. They influence tRNA structure, decoding, accuracy, and interaction with the ribosome. In this way, the tRNA modifications can fine-tune translation efficiency. The tRNA molecule, and here, for example, you can see one of the examples of the tRNA modifications, chemical modifications, which is sulfonylation, thiolation of the specific nucleotide in the glutamine tRNA. And inactivation of the enzyme that introduces this modification, the enzyme is called MNMA, leads to the embryo lethality in Arabidopsis, showing that the modifications of the chloroplast tRNAs are crucial for the proper growth and development of plants. But tRNAs are quite difficult to study because they are highly structured and heavily modified. However, we can analyze them using the optimized tRNA sequencing methods. In our work, we optimized a next generation sequencing-based tRNA sequencing approach to study chloroplast tRNAs. This tRNA sequencing method takes advantage of the highly processive reverse transcriptase, which introduces misincorporations and premature stop signals at the modified nucleotides in the tRNA during the cDNA synthesis. As a result, the method enables genome-wide profiling of both tRNA abundance and tRNA modification patterns. Here you can see the misincorporations that reflect the positions of the modified nucleotides across all chloroplast tRNAs, and these modifications are highlighted in blue and green colors. For example, the arginine tRNA shows a modified nucleotide at position 34, which is the first anticodon position, which is known from other studies to carry the inosine. As you can see, many chloroplast tRNAs carry multiple modifications, and importantly, we can distinguish the type of some modifications because the misincorporations are not random, but generate specific signals. For example, the inosine typically is read as G, while the M1G is often read as C or D. So we can implement this tRNA sequencing methods to both analyze the tRNA levels and also the modification status of the tRNA molecules. Based on the tRNA sequencing, we generated the map of the chloroplast tRNA modifications, which is shown in this figure. And interestingly, we identified both prokaryotic and eukaryotic type modifications in chloroplast tRNAs. An example of the prokaryotic type modification is ACP3U at position 47 in the variable loop in the tRNA. And this modification is not surprising because the chloroplast originated from the prokaryotes. So the prokaryotic type modifications are not very surprising to be observed in the tRNA molecules, in the chloroplast tRNA molecules. In contrast, the presence of the eukaryotic specific modifications, such as M22G at position 26, is more unexpected and may reflect the long coevolution of the chloroplast with the eukaryotic hosts. Importantly, we also observed the presence of the recently discovered novel cytosine modification called AVA2C at position 34, first anticodon position in isoleucine tRNA across diverse plant species. And this modification is crucial for the decoding of the genetic information by the tRNA. After establishing the tRNA sequencing protocol, we used it to analyze the effect of the highlight on the chloroplast tRNAs. And we observed that highlight is associated with changes in the abundance of 4 chloroplast tRNAs, which are induced at early stages of highlight exposure, but are repressed at later stages. Interestingly, we also detected changes in the levels of selected tRNA modifications, including the U47 modification in the lysine tRNA. And this modification is likely ACP3U that I mentioned on the previous slide. And we hypothesized that abundance of this modification increases during highlight exposure. However, this observation requires further investigation. Together, these results suggest that highlight reshapes the tRNA pool and its modification landscape to coordinate translation. Finally, I would like to briefly introduce the direct RNA sequencing using nanopore technology. This method sequences native RNA molecule directly and does not require the cDNA synthesis. However, the cDNA synthesis is usually implemented during the preparation of the library for the sequencing, but only the RNA molecule is sequenced from this hybrid cDNA RNA molecule. As a result, it preserves the RNA modification and provides long reads of the full-length transcripts. And importantly, at present, this approach is not yet optimized for the tRNA sequencing. However, recently there was a paper in Nature Biotechnology showing for the first time that the nanopore can be used for the transfer RNA sequencing. However, still, as I said, this method is not optimized because it generates quite a lot of noise. However, we used it to analyze chloroplast ribosomal RNAs, which also contain multiple chemical modifications. So we wanted to test whether this approach can be used to analyze the chloroplast RNA biology. Overall, I would like to say that this method, the direct RNA sequencing, opens a new possibilities for studying RNA regulation directly. And in recent work, we are still working on the data and planning to submit the manuscript shortly. We focus on 2 chloroplast ribosomal RNAs, 16S RNAs from the small subunit of ribosome and 23S rRNA from the large subunit of the ribosome. And in our experiments, we also included the in vitro transcribed rRNA molecules, which served as negative controls because they do not contain the chemical modification. And this negative control is crucial for the analysis because it allows us to compare the signal from the modified molecule, from the native molecule from the chloroplast to the molecule without chemical modifications. As a result, we generated the map of the chemical modifications in the chloroplast ribosomal RNAs, which is shown in this plot as the individual dots. And many of these modifications are conserved between bacteria and chloroplast. Again, it's not surprising because of the bacterial origin of the chloroplast in plants. However, we also identified a chloroplast-specific modification, only single modification in the 23S rRNA. Taken together, these results show that nanopore sequencing can be used to analyze RNA modification in chloroplast, and hopefully we'll try to implement this method to analyze the transfer RNA modifications. Here, I would like to summarize the main points of this presentation, and highlight creates a strong demand for the rapid PS2 repair. And as a result, chloroplast gene expression is regulated primarily at the level of translation. And translation is controlled at multiple stages, including initiation and elongation. The PSBA gene illustrates how translation initiation can be regulated through changes in the mRNA structure, while a ribosome posing adds another regulatory layer during the elongation. And our recent work further supports or shows that the tRNA abundance and tRNA modifications fine-tune the chloroplast translation. And we also, as I mentioned on the previous slide, are planning to implement the direct RNA sequencing to analyze the tRNA levels and tRNA modification. Interestingly, on the recent RNA biology conference, I saw the presentation showing that the nanopore can also be used to analyze the amino acid loading on the tRNA molecule, which also allows us to analyze this aspect of the tRNA biology. Together, these mechanisms enable rapid acclimation of the chloroplast to the highlight conditions. And in this slide, I would like to acknowledge all lab members who contributed to the work presented today. And in particular, I would like to thank Kinga Gołębiewska, who optimized tRNA sequencing method, which was not trivial work. And also, I would like to thank our collaborators for valuable contributions and discussions. And finally, I would like to acknowledge the funding agency, which is a Polish National Science Center that supported this research. And also, I would like to thank you for your attention. And I will be happy to take any questions. Thank you.
Okay, thank you very much for the presentation. That was really interesting. I learned a lot, and I have a lot of questions, and I see a lot of questions as well in the chat. So I will start taking the question from the chat with the first question from Julian Cuevas, who is working on dragon fruit. And he's mentioning that the excess of light damage this type of plant or fruit. And they have to use a special mesh to reduce the impact of light. However, he's wondering, how can we know the most appropriate level of shadowing? Experiments suggest between 20 to 40% shadowing, but mostly based on flower and damage, not on the level you presented. So do you suggest another approach to evaluate the good level of shadowing for this kind of crop?
Basically, the problem with the highlight appears when the light light intensity exceeds the photosynthesis of the plant. So probably I would check whether the photosynthesis is already saturated in specific conditions. And when the photosynthesis is saturated, and surprisingly, the saturation comes quite early at not very high light intensities, because, for example, in Arabidopsis, the saturation is at 500 micromoles of photons per square meter per second, which is like a regular sunny day, not in the middle of the day in summer, for example. And when the photosynthesis is already saturated, then the acclimatory responses must be induced to reduce the stress of the highlight. So probably I would check maybe the light intensity that saturates the photosynthesis in this particular plant, and try to adjust this level of the light intensity that already saturates the photosynthesis.
Okay. Yeah.
But I think the problem is that it's not on the leaf, but rather on the fruit, right?
He mentioned covering the plant with different mesh, flower buds, yes.
Flower buds. Oh, okay. I think also that the flower buds and also the very tiny leaves are also more sensitive to the light stress. We can often see this even in Arabidopsis, that is quite resistant to the highlight stress, to be honest. And usually because this is because the photosynthetic machinery is not yet established in these tissues, and thus the acclimatory responses do not work efficiently in young tissues. So
it depends on the maturation of the plant as well.
Yes.
Okay. Another question is, what is the role of the spacer in the accommodation of the mRNA?
Of the accommodation of the ribosome on the mRNA, I believe.
Yeah, I think
the spacer between the start codon and the Shine-Dalgarno sequence is crucial, to be honest, because the the optimal distance between these two elements, between the start codon and the Schneidel-Gardo, it's variable, but usually the optimal distance is 7 to 9 nucleotides, and it's very conserved, to be honest. However, it's also very interesting because some mRNAs in chloroplast and also in bacteria, about 30%, to be honest, lack the Schneidel-Gardo sequence. So not all of them actually rely on these mechanisms to induce translation. Yeah. Yeah. But in those that have the Schneidel-Gardo, the distance is usually, as I said, between 7 and 9 nucleotides upstream the start codon.
Another question is, does the ribosome pausing create some stacking loops in mRNA, or does it just stop the translation?
I think there are two different mechanisms. First is pausing, and it's temporal, so it pauses or slows down, and then it continues the translation. And another response is ribosome stalling, which I did not mention here. It's not analyzed in plants, but it's quite well understood in animal systems and yeast, for example. And this ribosome stalling causes the permanent stationary accommodation of the ribosome of the mRNA, and it actually is very problematic because the translation of the particular mRNA is completely blocked. And the ribosome is not active anymore, and the specific factors, the protein factors, need to disassemble the ribosome from the mRNA and usually degrade the mRNA. So those are the two different mechanisms. But on the other hand, there are also the situations of the ribosome collisions. So when we have the ribosome posed on a specific position on the mRNA, the next ribosome coming from the beginning of the mRNA can collide with the ribosome in the front. So those situations can also be observed, and also can be observed in the ribosome profiling data. But we believe, actually we believe that this posing is biologically relevant because it gives time to the nascent amino acid chain to fold, for example. I did not show it, but we also showed in the publication that it creates some time to fold, and also, for example, to anchor to the membrane, like photosynthetic membrane.
And just to build on this answer, what's the physiological response of the plant to this? Can it cause like toxicity effect because of accumulation of stuck ribosomal molecules on RNA, or?
We don't think so. We don't think so. We have not observed any toxicity, but honestly, we do not have the system to test that, because we can only try to describe whether this posing actually occurs on mRNA and whether it's somehow related to the biology of the nascent polypeptide chain or to the mRNA. But for us, it's quite difficult to modify the mRNA, for example, to introduce the artificial pose site to see the effect. And this problem comes from the difficulty to transform the chloroplast genome. And only one lab actually can modify the chloroplast genome of Arabidopsis. Is Ralph Bock laboratory, and it's very difficult to establish technology. We wanted to test that because it would be very, very nice approach, but we're not able.
Yeah, sure. Because one question that arises from your talk is, can we modify the plants to be more adapted to highlight exposure? Yeah. So
Maybe it's a naive answer, but I would say that the translation is optimized during the evolution to be as efficient as possible. So we can see only the picture where the posing actually is observed, but I don't know if there is any window for the improvement of this translation. It could be, but we don't know.
Okay, we should test this if we can modify one day the genome of the chloroplast. Yeah,
it is possible to do, for example, in tobacco. It's quite well known that the chloroplast genome in tobacco can be modified, and many things can be tested there. It's also very laborious, but still can be done. But in Arabidopsis is almost impossible. As I said, only a single laboratory on Earth can do it.
Okay, one challenge. I have another. There is another question. It's about the paper about the nanopore sequencing. So you mentioned a specific study on nanopore sequencing, and Patrick Vermeer is asking for the title of the paper, if you have it. Have information about the secondary structure by deducing it from the speed of sequencing.
Good question. I haven't seen any type of publications like that. It would make sense, right? Because if we have the strong secondary structure of the RNA, it will slow down the migration of the RNA through pore. But on the other hand, the cDNA is synthesized to unfold the RNA before the sequencing itself. There were the protocols that avoided almost completely the cDNA synthesis, but in our approach, we synthesized the cDNA just to unfold the RNA structure, and then the RNA strand was sequenced from this hybrid RNA.
So at the moment, it's not possible to sequence native RNA with its own conformation on structure.
Yes, exactly. We can see the modifications, the ends of the molecule, but not the native structure. I saw the publication implementing the nanopore sequencing, but on the chemical probing methods. As I mentioned during the presentation, we were using two different chemical probing approaches, the DMS and the SHAPE. It basically methods, the chemical is used to modify single-stranded RNA nucleotides, and those modifications can be read during the sequencing. And this is where the nanopore sequencing was actually used. And it has the advantage because the whole RNA molecule can be sequenced to analyze its structure. Not as in the Illumina sequencing, where we have to share into shorter pieces the RNA.
Yeah, obviously, Oxford Nanopore has a big advantage for this kind of.
And honestly, I was also surprised by the yield of the reads that were produced during our experiment, because our lab was looking at the nanopore sequencing opportunity some time ago, and the yield was very low. Usually from the single flow cell, it generated less than 1 million reads of the RNA, but right now we generated from the single experiment more than 6 million reads. So it was very efficient.
Yeah, we see the same with the new flow cell, the V10 version. Yeah, it has made a big change, right?
Yes, exactly.
I have another question that is a bit upstream from what you described today. So very impressive, the precision that you can get on the modification. But I wanted to know, do you have any indication about how the modification occur? So you have highlight, and then there are this modification, but what happened in between? What triggers the modification of tRNA? What triggers the modification of secondary structure? Do we have an idea about the transduction?
Good question. This is another branch of our research, because we are also working on the enzymes that intro these modifications. And this is just the start of this research, the beginning of this research, but we wanted to identify at least all the enzymes that introduce the modification in chloroplast tRNAs. We identified about 30 such enzymes, and right now we are characterizing them. At this point, we do not see the regulation of the expression of the genes encoding for these enzymes, but we cannot exclude the situation that the enzyme activity is also regulated by the conditions right now. But this will come in the future, I would say. Okay? Because right now we are trying to identify the enzyme that introduces the specific modification, and later on we will see whether it's regulated or not.
Yeah, well, that's very exciting. Also, you mentioned that the response is quite quick. Do you think that there is two phases? There is an acute phase of response and a later phase, which will lead to chronic adaptation, or?
If your question goes directly to the translation of the PSBA, I would say no. I think the response is constant. So it's related to the damage rate of the D1, and the translation efficiency depends on the, for example, light intensity. And the recent works actually from the Barkan lab also actually works a lot on the translation regulation of the PSBA, shows directly that the damage of the D1 protein leads or regulates the induction of the translation of the PSBA, which is also quite interesting. So the more damage we have, the higher translation efficiency we can observe. Okay.
Yeah.
But there is also another layer of acclimation, right? Because I was talking just about the D1 translation or the translation of the PSBA, but there are different layers of the acclimation related to the antioxidant enzymes or antioxidant metabolites and many different things.
Do you think that there could be an epigenetic footprint after exposure to stress? We would see permanent changes in chloroplast genome? Or?
This is a good question. We hypothesized that once because there were some works showing that the reactive oxygen species accumulation can lead to the modification of the RNA. And I think there is such a possibility because for example, the hydrogen peroxide is also produced during the high light stress, and it could potentially modify the RNA during the high light stress. So there could be some modification of the RNA.
Okay, very interesting. I don't see any question in the chat. Maybe I have another last question, maybe from the point of view of integrative biology. If we do a parallel with human biology, we can see that microbiome can lead to adaptation of skin cells to highlight, for example. Do you think that there could be a relation between microbiome and the effect you see in adaptation to highlight?
This is an intriguing question, right? Because I would say that our knowledge about the interaction between the microbiome and the plants actually it's very limited at this point.
At this point,
but for sure, there are some works showing that some pathogens, the plant pathogens, can influence the behavior of the chloroplast and also the translational machinery in the chloroplast. So at least since there is a lot of effort to study the plant pathogen interaction, there were some works like this. But on the other hand, there is also the question whether there would be the beneficial impact between the microbiome and the and the processes occurring in the chloroplast. I think it's a nice hypothesis to study in the future, to be honest.
Yeah, there could be a kind of crosstalk.
Yeah.
Yeah. Okay, a lot to do.
A lot of
a lot of things we don't know and a lot to do. Yeah.
True.
Since I don't see any question anymore, I would like to thank you again, Piotr, for the presentation. That was
thank you very much
highlighting
for the invitation, and also I would like to thank you for the questions.
And see you next time, maybe in Paris for the next scientific mission. And I would like to say to our audience today that just stay in touch with our website. We have a lot a lot of events ongoing, so register if you are interested. There are scientific missions, there are workshops, there are courses, so just come to us. So thank you again, Piotr, and have a good day.
Thank you.
And thank you
have a nice day
from our auditor. Bye-bye.
Bye.